Okay, so the first step of the cell cycle is the interphase. The interphase takes up most of the cell cycle, and this is the period where the cell gets ready for duplication. I mean, meiosis. It is divided into three steps: G1, synthesis, and G2, respectively. G1 is the starting point. The cell begins carrying out regular tasks. However, it is growing in size and duplicating the organelles necessary for meiosis. The next phase is called synthesis. This process focuses on replicating DNA. And all the cell's processes cease because it only focuses on replicating DNA. The cell will also begin making identical copies of DNA. The next phase is G2. The cell once again prepares for cell division by creating more cytoplasm. Many other important organelles such as the mitochondria are replicated. This phase continues until the cell enters meiosis. Chromosomes.
The end result of meiosis 1 is the separation of homologous chromosomes. In the beginning of prophase 1, the chromosomes begin to condense and homologous chromosomes connect to each other, which is called synapsis. During synapsis, crossing over occurs, meaning that each pair of homologous chromosomes mix their genetic material, which results in a genetic rearrangement.
Oh, sorry, we were signaling you for like 5 seconds. Who signaled? No one signaled. Okay.
Yeah, can you please do that?
A charismat exits at the point where crossing over occurs and holds the two original sister chromatids together.
So they're held together.
There is also centrosome movement. The spindle formation and the nuclear envelope break down.
And the nucleus
Just breaks down. The last part of prophase 1 includes the homologous pairs moving to the metaphase plate. In metaphase 1, the homologous chromosomes are at the plate and are attached to connectotroids, microtubules. Sorry.
Don't attach to the middle, attach to like the middle piece. So it's like, I'll take it off.
Yeah. And the polar forces between the microtubules just keeps them in place. Mm-hmm. And just a fun fact, it's one of the straightest lines in all of nature.
These are the actual ones.
In anaphase 1, the homologies separate and move to opposite ends due to the spindle fibers, but the sister chromatids stay together. In telophase 1, each half of the cell has a complete haploid set. Cytokinesis in the division of cytoplasm and results in the two haploid daughter cells.
The nucleus also forms in the telophase. So new nucleus form for each of these things. And then the cell becomes two cells. And this concludes meiosis 1.
During meiosis and then telophase and cytokinesis,
Move back. Okay, go.
two haploid cells form, and a haploid cell can be represented as N because it is the number of chromosomes that is needed to form a complete diploid cell. An example of this would be a human. The haploid number in humans is 23, and 22 autosomes in one single sex chromosome. In meiosis, a diploid cell separates to form a haploid cell. Your father would have 23 chromosomes, and your mother would have 23 also. In one cell forming a diploid, The father and mother's chromosomes will duplicate and will split into haploid cells, and the sister's chromatids will separate into haploid cells with unduplicated chromosomes.
Meiosis 2. In prophase 2, a spindle forms. In late prophase 2, chromosomes each still comprised of 2 chromosomes associate at the centromere after the nuclear envelope dissolves. And they move towards the metaphasic 2 plate. Metaphase 2, the chromosomes are positioned at the metaphase plate as in meiosis 1. Because of the crossing over in meiosis 1, the 2 sister chromatids of each chromosome are not genetically identical. The kinetic cores of the sister chromatids are attached to microtubules extending to opposite poles. In anaphase 2, breakdown of proteins holding the chromatids together at the centromere allow for the chromatids to separate. The chromatids move towards opposite poles as individual chromatids. Telophase 2 and cytokinesis, nuclei form. The chromosomes begin decondensing and cytokinesis occurs. The meiotic division of one parent cell procures daughter cells, each with a haploid set of unduplicated chromosomes. The 4 daughter cells are genetically distant from one another and from the parent cell.
No, we're gonna stop the video or something. Wait, then we're good. If we stop the video at the right time.
And they die after.
These are the 4 haploid cells created through meiosis 2, and as you can see, the chromatids have decondensed into DNA.
In the end of meiosis, the division produces haploid daughter cells with half the genetic material of the parent cell. Through the crossing over that occurs in prophase 1, the combination of chromosomes is different, which results in chromosomal combination different from the parent cell. This is important because the difference of daughter cells not being identical to the parent cells and having to combine through fertilization and the recombination leads to genetic variation, which is important for the survival of life.